सर जी आप आ जाओ, अपना मेल खोल दो। आ गया आ गया।
नीचे मैं ऑन कर लेता हूँ।
ये है।
यहाँ पे क्यों नहीं आ रहा?
पेन हो जाएगा सर, वही रहने दो पॉइंट उसकी बल।
नहीं मैम, ये नहीं होगा। मैम माउस का फिर वही दिखाना पड़ेगा, एरो से। है नहीं इसमें।
1
हेलो। 170। 170। एक मिनट बस। Okay, sir. Start. Good morning, everybody. Today I'll be presenting my doctoral work titled as Elucidating the Mechanisms of Gallstone Formation and Genetic Landscape of Gallbladder Cancer. So the outline thesis will be talking about functions of gallbladder rationale for designing study based on in literature, aims, objectives, and then talking about results such as geobiology of gallstones, development of animal model of gallstone disease, genetics of gallbladder cancer, study of cancer evolution using patient-derived xenograft mouse model, and then limitations and way forward. So first and foremost question, why even study gallbladder? Because it is a very important organ and it also helps in storage of bile and also concentration, which protects the colon and intestine. It also protects the liver from the harmful effect of secondary bile acid. Now, gallstone is one of the most common disease of gallbladder, which occurs due to supersaturation of bile with the cholesterol, and some of these cases lead to development of gallbladder cancer. The prevalence of gallstone is very high in South American countries such as Chile, but is also very high in India even, and the prevalence range from 4 to 9%. According to the recent Tata Memorial study, it is high in North India and Northeast of India compared to South of India, and various risk factors include obesity, pregnancy, and diet are associated with gallstone disease, and is much more prevalent in female compared to male with a ratio of 3:1. The gallbladder cancer is very high in Asian countries. Almost 70% of total global cases come from Asia, and India is showing one of the most highest cases of gallbladder cancer with an incidence rate of 22 per 100,000 individual. Now, the chronic cholecystitis or inflammation from the gallstone lead to the tissue transformation of gallbladder epithelium into the pre-neoplastic situations such as metaplasia, dysplasia, ultimately forming the carcinoma. But 10 to 20% of gallbladder cancer cases arises in the absence of gallstone. And from the literature, we know that there is a role of microbial infections, specifically Helicobacter and Salmonella typhi in the gallstone formation. And there is also role of immune system, specifically neutrophil extracellular traps from neutrophils, which act as super glue, which binds the cholesterol and start the gallstone initiation. So both of these microbial and immune system start the inflammatory nexus leading to development of gallstones, and the persistent irritation lead to DNA damage, oxidative stress, ultimately forming the gallbladder cancer. Based on the literature, there were some gaps, such as there was no data about the long-term impact of gallstone residence time on the risk of development of gallbladder cancer. There was absence of non-invasive biomarker that can predict gallstone disease and gallbladder cancer, and scanty data about divergence that are frequently mutated in Indian population in gallbladder cancer. Based on these, some key questions were asked. Is there any impact of gallstone in duration and its constituents on the disease progression? Can we develop a metabolite-induced mouse model? And what is the genetic landscape of gallbladder cancer, specifically in North Indian cohort? And are there any difference in cancers that arise in the presence or absence of gallbladder? So based on these three key objectives were designed. First one was physicochemical characterization of gallstones using carbon dating, 6ns rRNA sequencing, and metabolome analysis. A part of this study has already been published. The second objective was to development of diet-based metabolite-induced gallstone disease mouse model. The third one was to identify the genetic landscape of gallbladder cancer and then correlating with already existing immunohistochemical-based prognostic markers. And the study was designed in both prospective and retrospective cohort. In prospective cohort, healthy individuals, gallstone disease patients, and gallbladder cancer patients were enrolled. Blood, bile, stone, and tissue were enrolled wherever was feasible. From blood, PBMC was isolated and Sanger sequencing was done for LPEC associated stones such as ABCB4. Plasma, bile, and gallstone were subjected to metabolome analysis. TMA pathways identified and a mouse model was developed. From stone, carbon dating was done to reduce its age, duration, and 6s rRNA sequencing was done to reduce its microbial components. From tissue, a mouse model was developed, patient-derived xenograft mouse model was developed, and from their genetic evolution of the cancer was studied. In the retrospective cohort, since we got limited number of fresh tissue samples from gallbladder cancer, so tissue samples were collected from histopathology, and then next generation sequencing was done for a targeted panel of 42 genes of the LBS. So coming to objective one, which is about physicochemical characterization of gallstones using carbon dating, 6S rRNA sequencing, and metabolome analysis. So limited data is known already, such as there was only one study which showed that the longer retention of gallstone might be associated with increased risk of cancer. There is almost all the studies that study the chemical composition of gallstone use FTIR, spectroscopy based technique FTIR, but it's limited and only give a handful of insight into the chemical constituents. There is already known microbial community that are known, such as Helicobacter and Salmonella with the gallstone. So based on this study was designed, since I was new to this study, I decided to do a pilot study first to test the feasibility of doing carbon dating and all these techniques to gallstones. And this study has already been published in Journal of Gastrointestinal Surgery. But this study has several limitations, including small sample size, absence of gallstone from gallbladder cancer patients. So a new study was designed, and the patients with chronic cholecystis only were classified as GSD or gallstone disease. Metaplasia and dysplasia were clubbed together as PNP or preneoplasia, and gallbladder cancers were classified as GBC. And then these patients were subjected to radiocarbon, these stones were subjected to radiocarbon dating, metabolome analysis, and 6S rRNA sequencing. So this is a representative graph of how does a gallstone normally looks like when we chop it. So from the core layer of the gallstone and outermost layer of gallstone were taken, and then they were subjected to radiocarbon dating and to deduce its actual age of formation. Based on the age of formation of core layer and date of surgery, the total duration of gallstone inside the gallbladder was deduced, and we found that the benign gallstone or chronic cholecystis gallstone has a median duration of 6 years, compared to preneoplasia of 9 years and highest in GBC, which was significantly high compared to benign gallstones. So to conclude, GBC patients exhibited a significantly higher gallstone residence time when compared with the benign gallstone. Next, to know about the constituents of the gallstone, we did it in two parts. First, to know about the chemical composition, and another in the bacterial composition. For the chemical composition, first we did the standard biochemical analysis, which suggests that all of the gallstone that we had were in high cholesterol and low phospholipid. To know about the chemical composition, first we did FTIR in the pilot study, but it gave only limited insight into the chemicals such as cholesterol, calcium carbonate, calcium bilirubinate. But most of these compounds were inorganic and limited organic. So to overcome this limitation, we decided to do LCMS in our current study. For bacterial taxa, we did 6S rRNA sequencing post confirmation of presence of microbes from scanning electron microscopy. The core of the gallstone was then subjected to metabolome analysis to specifically identify metabolites that might be present during the genesis of gallstone. And the PCA plot shows the clustering of different samples in different groups, which also show the distinction among different groups. The representative heat map shows the relative abundance of different metabolites, such as these, as you can see here, are highly enriched in gallbladder cancer associated stones, and these are significantly down-regulating gallbladder cancer associated stones. Most of these metabolites were either in lipid category, bacterial metabolites, or bile acids. Then these were subjected to differential analysis, and the metabolites that were significantly up or down regulated were divided. And in upregulated metabolites, we found metabolites such as 20-carboxy LTB4, which is non-inflammatory in nature, secondary bile acid, sulfoglycolic acid, and bacterial or host dibacterial metabolites such as TMA were significantly enriched in gallbladder cancer associated stones. In the downregulated metabolites, we found plasmamembrane associated lipids downregulation such as phospholipids and lyophosphatidylcholine. There is also downregulation of non-bacterial metabolite or short chain fatty acid, butyric acid, which is non-protective in nature, and also decrease in chloroquinodeoxycholic acid. Since we found multiple bacterial metabolites, we asked question: what if can we found any bacterial taxa that might be associated with these metabolites, and is there any change in these? So for that, we did 16S rRNA sequencing, and the PCO plot shows the difference among these groups based on the clustering of different samples. The alpha diversity index, specifically using Shannon, shows the decreased diversity, although the difference is non-significant. The differential analysis was then done to find the abundant genus in both gallbladder cancer associated stone and GSD, and we found that Likely pathogenic bacteria such as Klebsiella and Enterococcus were specifically increased in gallbladder cancer associated stones, while all other genus except Escherichia were more increased in benign gallstone disease. So this conclude that the cancer-associated gallstones shows reduced diversity but increased abundance of pathogenic Texas Klebsiella and Enterococcus. So to conclude, the gallstone residence time of more than 12 years with metabolites such as sulfoglycolithocholic acid, leukotriene B4, TMAO, other bacterial tagatronic acid, and microbial species like Klebsiella and Enterococcus might be associated with progression of gallstone disease, benign gallstone disease towards the gallbladder cancer. While lower duration of less than 12 years with primary bile acid, deoxycholic acid, butyric acid, Enterobacter, and Streptococcus might be preventive in nature. Since we had found gallstone, TMAO in the gallstone, we also did metabolome analysis in the bile. I am not showing the data right now. And we found that the precursor of TMAO, which is TMA, trimethylamine, was found to be significantly increased in gallbladder cancer associated bile samples. So since we were getting TMA pathway, we asked question, is there any relevance of this pathway in our disease pathology, which is gallstone? TMA, which is forming in the gut from its precursors, choline, carnitine, betaine, and it comes from the dietary products such as red meat, fish, and even also in the vegan products, beans, peas, canola oil, mustard oil. It is known that it gets transported to the bile, to the liver, where it gets converted into TMAO with the liver enzyme flavin monoxidase 3. And TMA is known to have impact on the cardiovascular disease, atherosclerosis. And there are published papers also which show that TMA is associated with fatty liver disease, PSC, and there is one published study also says its role in gallstone formation. So since we had found metabolome TMA and TM in both gallstone and bile, we asked question, what about the levels of TM in plasma for gallstone disease, and can a dietary supplementation of TMA can induce gallstone disease in animal model? So based on this, second objective was design, and in this we identified plasma metabolites and developed a animal model. In this again, 170 individuals were selected in both discovery and validation cohort. Plasma sample was isolated and was subjected to untargeted metabolomics. And the biomarker which is TMAO, trimethylamine oxide, was identified, and this was further validated using targeted metabolomics to find out the actual concentration of TMA in the plasma sample, and a mouse model was developed. This mouse model and the tissue from these mouse were taken for gene expression. And then the plasma metabolomics from the discovery cohort shows again the PCA plot shows association of different samples, and which also show the distinction of these samples across different groups. These metabolites were then subjected to volcano plot analysis to identify differentially regulated metabolites with folds in more than 2 and p-value less than 0.05. From all those differentially regulated metabolites, we wanted to find metabolites that are specifically that can be used for both for prediction of gallstone disease and gallbladder cancer. So the common 20 metabolites were taken, and these common 20 metabolites were taken that were specific for both gallstone disease and gallbladder cancer. The representative heat map shows the relative abundance of all these metabolites, all these 20 metabolites, and TMA was found to be significantly increased in both gallstone disease and gallbladder cancer. Then the 20 metabolites subject to a AUC curve analysis, which shows that the signature of these 20 metabolites shows high accuracy of 0.98 for GSD and 0.93 for gallbladder cancer. Besides this, we also try to identify the pathway that might be associated with these 20 metabolites, and two pathways that were significantly and highly increased were flavin monoxidase oxide nucleophiles and diet-dependent TMA trimethylamine and TMAO trimethylamine oxide metabolism. Since we found 20 metabolites, we asked question, can we find one specific metabolite that can actually help in prediction of gallstone disease and gallbladder cancer? TMAO shows high AUC of 0.94 for gallstone in discovery cohort and slightly lower but still good 0.83 for validation cohort. And also for the cancer, the accuracy was slightly lower than compared to GSD, which was 0.7 for gallbladder cancer and 0.62 for gallbladder cancer. All this data was based on untargeted metabolomics and specifically based on the relative abundance. So next we ask question, what about the actual concentration of TMAO in the plasma? So for that we bought the standard of TMAO and found out the actual concentration of TMAO in the plasma. And we found that GSD and CAGB, the patient from GSD and CA gallbladder cancer shows high concentration of TMAO in the plasma samples, which was very high compared to healthy control. So since TMA is already present very high in GSD and CSG, we ask question, can TMA supplementation will influence the Gallstone disease in mice? So we fed high cholesterol diet, which is lithogenic diet with TMAO, and look at the impact of these on the Gallstone formation. So for that, a mice study was designed. Mice were grouped into divided into three groups: chow diet or a control diet, lithogenic diet plus 0.3% TMAO. The mice were sacrificed at intervals 3 and 6 months, and their diet consumption, body weight, glucose level, and the oral glucose tolerance test was done at both 3 and 6 months, although no significant difference was observed. When mice were sacrificed at 3 and 6 months, we found gallstones had only developed in lithogenic diet plus TMAO at 3 months. At 6 months, although gallstones had developed in 3 months at a lithogenic diet group and also in lithogenic diet plus TMAO group, but in lithogenic diet plus TMAO group, we found a metaplastic change in the gallbladder epithelium. So this concludes that TMA not only accelerated the gallstone formation or also induced metaplastic change in the gallbladder epithelium. Next, we wanted to look at the gene expression profile that might be changed due to this TMA intake. So we checked mRNA expression profile of various transporters, including cholesterol, phospholipid, bile, water, also anti and pro-inflammatory markers. And out of all these genes, we found significant difference in only in these four of genes, which are TGR5, ABCC3, which are known bile acid transporters. The master regulator of bile acid signaling FXR was found to be significantly upregulated, while anti-inflammatory marker IL-10 was found to be significantly decreased. So to conclude, TMA, which was getting formed into the gut from the dietary intake of carnitine, choline, glycine, betaine, and it was getting transported into the liver, and which was getting converted into TMA using the help of flavin monoxidase 3. This TMA was somehow regulating the expression of these four genes, FXR, ABCC3, TGR5, IL-10, in the gallbladder epithelium. Due to decreased expression of ABCC3, there was increase in bile acid retention. Due to TGR5 decrease, there was reduced gallbladder motility, which lead to the cholestatic situation and lead to development of pro-inflammatory situation due to development of gallstone formation. In earlier paper, it was suggested that FXR might be associated with the dissolution of the gallstone, but in our data, we found that the increased expression was associated with increased gallstone frequency. Since we had studied the microbes and the metabolites, next we decided to study the key component of to study the cancer part, that is genetics. So we designed third objective, identifying the genetic landscape of gallbladder cancer using FFPE tissue and identify any familial link subject to gallstone. Besides that, I also did two more studies. First one was we did in families of the patients in which the gallstone was running in families. So for that, we enrolled only 6 families, and we checked the germline polymorphism in ABCB4 gene, which is ABCB4 gene, which is known phospholipid transporter. In these 6 families, we found mutations in only 2 families, which is one is already known, which is in the RS number 185 SNP site, while 2 novel heterozygous mutations were also found. For the somatic mutation analysis, we did next generation sequencing for 32 patients, and these were taken from the FFPE tissue samples. We also developed a patient xenograft model from the rare subtype of gallbladder cancer, adenosquamous carcinoma, and we studied the cancer evolution using that patient. So I'll be discussing these in further. So from the somatic mutation data, 3 molecular subtypes were identified based on the mutation clustering. GBC1 or cluster 1 had mutations in P53 gene along with oncogenes and tumor suppressors such as RB1 and KRAS and PIK3CA, while GBC2 shows mutation in P53 along with mutations in only RET and IDS1 and IDS2 genes, while GBC3 shows wild type P53 but oncogenic addiction in almost all other genes. So these 3 molecular subtypes shows almost similar age, the mean age was almost similar, but GBC2 shows high gallstone frequency when compared with the GBC1 and GBC2. So next we ask question, is there any association of these these different molecular subtypes based on genetic mutation with the survival? So when we developed the survival plot, we found that GBC3 was comparatively better when compared with GBC2 and GBC1, although the difference was non-significant. Next, we ask question, is there any difference in the gallstone that gallbladder cancer cases that arise in the presence of gallstone or in the absence of gallstone? So the patients were divided either which has a history of gallstone or without any history of gallstones, and we found the patients of gallbladder cancer patients with a history of gallstone had mutations in IDH1 gene, while the patients without underlying gallstone had more mutations in BRAF and MEP2 kinase 1. Besides that, we in these our data we also found known variants which are already actionable, and drugs could have been used for these patients for these specific variants. So to conclude, the gallbladder cancer cases that arise directly from the normal gallbladder had increased mutations of BRAF mutation MAP2 while gallbladder cancer with gallstone had increased IDH1 mutations and 3 molecular subtypes were identified. The wild type P53 had increased stromal podoplanine. I have also done IHA. I'm not showing the data due to time constraint. Had increased EGFR PI3K, was more prominent in male, had large tumor size, and the survival was poorest of only of 13 months. While the mutant P53 with EGFR and KRAS had median survival of 23 months, while P53 with RET and IDH1 had more Gallstone frequency and had the highest survival of 27 months. Next, the second subtype is to study the clonal evolution of gallbladder cancer using the patient-derived xenograft mouse model. So, a patient of adenosquamous carcinoma came to ILBS after undergoing cholecystectomy at another center. The tumor had spread to the omentum, and the tumor has spread to omentum. The patient underwent chemotherapy and then metastatectomy. The tissue from the metastatectomy was taken and then was injected into node skid mice. A tumor was developed after 4 months, and the tumor was harvested. So the tissue sample was from this tumor part, from metastatectomy sample, and even from the primary was taken. And since we had 3 different samples over a period of total 2 years, we decided to do somatic mutation analysis to check the genes that might be evolved during this course of 2 years. So first thing we did to confirm that all these 3 tissue were of same origin and also the adenosquamous origin. For that we did IHC with CK7, and to confirm the adenosquamous origin we did IHC with P63. To confirm that the tumor that was developed in the human in the PDX patient in the mice was of human origin, we did genetic analysis using human polymorphic marker which is MBOT in our case. From the genetic mutation study, we found that the pathogenic variant G12V of KRAS was conserved across all 3 samples, while LRP1 had benign mutations but also conserved in all 3 samples. Besides that, genes such as FGFR3, TERT, and RB1 were only present in the primary tumor, while genes like PIK3C, which are known to associate with cell survival and growth, were only present post chemotherapy and only in the metastatic tumor, while chromatin remodelers such as ARID1A, ARID2, and BEP1 were only present in the patient-derived xenograft model. Since we know that these genes are present, we decided to do the clonal evolution study. How actually these clones survived from the time of initiation of Gallstone up to the patient-derived xenograft. So we studied the clonal evolution using the already existing pipeline of our Cyclone. P.WEF represent the primary variant allele frequency of the primary tumor, that is the first one. R.WEF represent the recurrent variant allele frequency, which include both metastasis and the PDX tumor. The primary tumor had increased mutations of high frequency of LRP1B, KRAS, and RB1, but as the tumor developed in both metastasis and PDX, the variant allele frequency of different other genes such as ARID1A, ARID2, and BEP1 was almost similar to the initial mutation clones. So in the clone 1, which was supposed to be present at the initiation of Gallstone, had mutation in LRP1B and KRAS, and further subclone did develop, but the most mutation were present in the initial clone only. But in the case of recurrent tumor, that is metastasis and PDX, this primary clone further sub-evolved into another clone which had more mutation in ARID1A, ARID2, and BEP1 with more frequency of almost 95%. So to conclude, the initial clone for both primary and recurrent tumors had mutations in LRP1B, KRAS, and RB1. Multiple subclones were evolved from there, but the final clone had the mutations in ARID1A, ARID2, BEP1, in addition to already existing LRP1B, KRAS, and RB1. So to conclude, the PDX model that we had developed had resembled the primary, recapitulated the histological and molecular features of both primary and secondary. There was loss of RB1 in the primary driver and activation of PI3K in the secondary, and also in the PDX model there was activation of chromatin remodelers. So to conclude this thesis, the Gallstone that were developing from the normal gallbladder had duration of less than 12 years. There were more mutations in ABCB4 gene, and there were metabolites that TMO is associated with these gallstones. In gallbladder cancer development from gallstone patients, there were mutations in P53. Duration was gallstone inside the gallbladder more than 12 years. There were genetic mutations associated with IDH1, RB1. Multiple bacterial constants like Klebsiella and Enterococcus were associated. In multiple metabolite dityrosine, leukotriene, TMA, targatronic acid were increased. These TMA was then fed into mouse, and a mouse model was developed, which shows accelerated gallstone formation, metaplastic changes. While the PDX that we developed from the rare subtype of cancer of adenosquamous carcinoma was helped to study the clonal evolution. Besides that, the cancer that are forming directly from the normal has increased mutations in KRAS, LRP1B, BRAF, and MEP2 kinase. While we studied several limitations, including small sample size, and we only got a few intact stone from the gallbladder cancer patients. 16S rRNA sequencing revealed only a limited genus only, and we were not able to pinpoint the exact bacterial species that were driving the gallstone formation. TMO supplementation lead to progression into up to metaplasia, and limited gene of somatic panels provide only a restricted view. Now, way forward, we would love to do the shotgun metagenomics to accurately identify bacteria at species and strain level that actually drives the genesis of gallstone and gallbladder cancer, and then we can validate these bacterial and metabolite species in the mice model. Development of long-term animal model of TMAO for evaluating carcinogenicity for more than 6 months. Whole exome sequencing will help us further enable us to subclassify the genetic subtypes. The translational accept include TMA or sulfoglycolic acid to stratify gallstone disease for high risk group and screening for p53 driver mutations in cell-free DNA. I want to acknowledge my supervisor, Dr. Gayatri Ramakrishna, and my co-supervisors and all the HPV surgery team for providing me with clinical samples, and lab members, trainees, and CSIR providing fellowship, and to all the participants who gave their consent, also the animals that were sacrificed during this thesis. Thank you.
Yeah, so thank you, Vipin. And because of the time constraint, he could not present all the data, and he went like a super fast express trying to finish up within 25 minutes time limit. So I need to thank Professor Serene and Director Sir, because he was the one who gave us a gentle nudge and a snot so that we can take up something which is rarely discussed in this podium, because liver is the main focus.
Dr. Gayatri, we can have time for 5-10 minutes for questions.
Yes, so
our chief guest is yet to arrive, so
yeah,
we
so,
have some questions.
so we were actually, so there were too many things to be done. And the whole idea was to actually start with the genetics. But we thought that besides the genetics, there's a lot of material in the form of the gallstones. So he could do the chemical characterization, although he said that there were a lot of limitation. The limitation was getting an intact gallstone from the cancer post-surgery. And many a times we also found it was interesting, the patients did not want to part with them. So they would actually take away. So he ended up having only some 12 to 13 intact gallstones, of which few failed in our analysis. So he started with a pilot study and he showed already the data. So I would like to get the inputs, because now he's almost finished his thesis and his viva is also due next week. So I would be really happy if you can give some comments so that the next student who comes and joins can improve the work.
Anyhow, I think it's a very good work, and I think had you got more samples, it would have been, carried forward. I hope it is carried forward for further analysis. Any comments from anyone, or
Rakhi, you want to comment something?
or from the surgeons? So because this question comes quite often, people with gallstone disease, asymptomatic, do we get the surgery done or not done, get it done or not done? Yes. They remain asymptomatic for years together. So.
And also from the surgery department, if Dr. Ragini can comment on.
Yes, Anil, you wanted to say something?
You can start.
You wanted to say something?
Regarding the implantation. Yes, sir.
So I would say that it is a wonderful work, but my question to you is, how do we take forward from here? Whatever data you got, how do we take it into our clinics? This whatever nice work you did. So the genetics is one part, which I feel is important, the gut microbiota you're trying to show. But next part of this study, which I feel will be very important, is to look at whatever you found as associations, whether these are predictions. So the chicken and egg story kind. So it's like very important for us to know what is driving something, whether the cancer is driving the changes in the gut microbiota. The gut microbiota is driving the changes, and the epigenetics and those things are very important for us. What down regulates the transporters for these patients to develop gallbladder cancers. That is what I.
So
Would you like to comment on that?
No, let him comment because it's his thesis. Let him defend
So
first.
from the genetic study, I would say if we can get more sample that we can stratify the patients based on the survival, but that's post surgery only. And about the TMO association that we shown, if we can do it in almost 500 or 1,000 more patients, that we can actually find a risk score based on the TMO, and then we will be able to stratify the patients of the gallstone disease that might be able to progress towards the cancer. So we can take a better look at those patients, and we can focus on those patients that are supposed to be developing cancer from the gallstone disease. And from the first part, I can